slide we will observe. Then we will look at vas deferens, and then we will observe prostate. Now on the screen you see the testis epididymis slide. In the lowest magnification, I can see both different tissues. At the right side you can see the testis. These are seminiferous tubules. And at the left I see the epididymis. This is ductus epididymis. As you know, ductus epididymis is only one single duct, but it's so too many times convoluted surrounding the testis. Therefore, I can see many sections of the ductus epididymis here. How can I distinguish between two? It's not difficult. As you can see, in the testis, there are spermatogenic cells. The seminiferous tubules are filled with spermatogenic cells, and between the seminiferous tubules we can see the interstitial cells, the vessels, and the interstitial cells. And a thick capsule, fibrous dense capsule, tunica albuginea, is covering the testis. At the left, ductus epididymis has a large lumen and it's filled with sperm, because this is the storage site of sperm. Therefore, I can see that many sperm are here, filling the lumen of ductus epididymis, and it has a thin pseudostratified columnar epithelium. Now let's start with testis, and we observe one of the seminiferous tubules to see our germinal cells, the spermatogenic cells. Here is one seminiferous tubule. One, another one, and another one. At the base of the tubule, there are spermatogonia. Spermatogonia can be distinguished by being at the base of the tubule first, and also with round oval nucleus here. These are spermatogonia. Here I can see another one. And at the base of the tubule, I can also see the nucleus of Sertoli cell. I cannot distinguish the Sertoli cell cytoplasm borders, but the nucleus is like a triangular shape, and a distinct nucleolus can be seen. Here is another Sertoli cell nucleus, which is triangular shape, and a nucleolus can be seen in the middle of it. Here is another one with a triangular, clear nucleus and a nucleolus. And after spermatogonia, there are meiotic cells, primary spermatocytes. And in this tubule, these are these cells here. Very big cells, because they are replicating their DNA here. Here is the nucleus and the cell. They are replicating their DNA, therefore they are very big, and I can distinguish them easily from the spermatogenic cells at the base and Sertoli cell. And here is the primary spermatocyte. Then there should be the secondary spermatocytes, but we cannot observe them in most of the seminiferous tubule sections because they complete their meiotic division so fast. Therefore, I see the round spermatids here. The secondary spermatocytes fastly form round spermatids, and these are round spermatids. These are haploid cells with their round nucleus and cytoplasm around. And these are the tails of the sperm. And in this tubule, with round spermatids, also I can see the elongated spermatids, where their heads are embedded in the, between the cytoplasm of Sertoli cells, and their tail is extending to the lumen. And between there is the interstitial area. And in interstitial area, we can see round Leydig cells with acyclic cytoplasm and round nucleus, mostly eccentric, not located in the center but periphery. Here we can distinguish. So these are the cells we want you to observe today. Here is a very nice Leydig cell. This is the interstitial area because there is a vessel here. You can see a Leydig cell very, very well. The eccentric nucleus, it's not located in the center, and the acyclic cytoplasm. So the Leydig cells are in the interstitial area, and the Sertoli cells and the spermatogenic cells, spermatogonia, primary spermatocytes, round spermatids, and sperm are inside the seminiferous epithelium. Here is another Sertoli cell nucleus. It's triangular and the nucleus is prominent. And so if we leave testis and come here to epididymis, the epididymis, ductus epididymis, has pseudostratified columnar epithelium with prominent stereocilia. Here we can see the stereocilia of these epithelial cells. And inside the ductus epididymis, we can observe sperm cells. You see, this is the pseudostratified columnar epithelium, and these are the stereocilia extending through the lumen. And that you should see in this slide. Now, the second slide is ductus deferens or vas deferens slide. This slide, when we look at the lowest magnification, ductus deferens is also a single tube, single duct. And you can easily distinguish it with its very thick muscular layer. There is a very, very thick muscular layer. This is the epithelium, pseudostratified columnar epithelium with stereocilia, like epididymis, and it has not straight but a little bit convoluted, irregular epithelium, and the thick muscle layer is very distinct. There is a first layer, longitudinal, then comes the inner circular and the outer longitudinal again. And this is tunica adventitia, the serosa surrounding the vas deferens. So you can easily distinguish ductus deferens or vas deferens with its thick muscular layer, and here is the epithelium. Our last slide is prostate. Remember, it was the largest accessory sex gland. When we look at prostate, we will see first tubulo-alveolar glands. There are many tubulo-alveolar glands. This is one of the glands, another one, another one, another one. So the epithelium of these glands are also pseudostratified columnar epithelium. The secretion of prostate can be seen in the lumen as pinky stained. And another structure you should know that between these tubulo-alveolar glands there is a fibromuscular stroma of prostate. This is the fibromuscular stroma between the tubulo-alveolar glands. And a fibroelastic capsule is surrounding the prostate gland. And inside the gland, sometimes, here I cannot see, but sometimes you can see concentrations of these secretions, like calcifications we said, and they are called the prostate stones or corpora amnesia. Corpora amnesia. So I want you to observe the prostate glands, tubular glands, the fibromuscular stroma, and the fibroelastic capsule surrounding the gland. You can now work